into the classification of smooth muscle. So smooth muscle is classified into single unit smooth muscle and multi-unit smooth muscle. And the main difference is how the muscle behaves. So multi-unit smooth muscle, each one of the cells acts independently, okay? They are autonomous, they have their own function, they regulate their own function, and they're not acting as a unit. They're acting as independent cells that can be contracting or relaxing independently. There's also a difference between the way that the neuron interacts with these multi-unit cells. So essentially, the neuroeffector junction is going to exist between each and every single one of these cells. So the neuron is going to go deep throughout the entire muscle, and each cell will receive its own neurotransmitter and will have its own regulated function depending on that neurotransmitter and that neuroeffector synapse happening. With single unit smooth muscle, we see a different orientation. So the muscles are acting as one single unit. They are connected by gap junctions. So we don't have gap junctions in multi-unit. We do have gap junctions in single unit. And because of these gap junctions, these provide a means of communication where cells can speak to each other. So one cell can tell another cell, hey, this is what's going on, this is what we need to be doing. The other cell can tell its neighbor, and all of them can kind of be on the same page and be doing the same thing. I like to think of this as the cells kind of holding hands. They're singing from the same, singing from the same hymn, if you will. They're doing the same thing, they're communicating, and so they're acting in unison, in cohesion with one another. Because of that, they don't need to be receiving the same autonomic stimulus independently. The cells at the very top layer can receive that stimulus, so only the cells at the top layer here are receiving neurotransmitter input from the neuron, and then they will just communicate through each other via the gap junctions what that neurotransmitter is doing. So in this way, single unit smooth muscle can behave like a single unit, whereas multi-unit smooth muscle, the cells are acting independently and are receiving their own signals and doing their own thing, right? Contracting or relaxing independent of each other. So let's talk about kind of the locations, some examples, and more features of single unit, and we'll do the same for multi-unit coming up. So single unit is the most common type, most abundant throughout the body. Common locations are the intestinal tract. We also see them lining our blood vessels. We also see them lining the respiratory tract in some areas. And these are activated synchronously. So once we activate or depolarize that top layer, the impulse can spread throughout the entire muscle because of the gap junctions and these cells communicating to one another. All right. So they will contract as a single unit when that neural signal is received. Single unit smooth muscle, other features here, they have gap junctions, which you mentioned previously. They have pacemaker ability, so they can spontaneously depolarize. And just to kind of go a bit further about what this means, we talked about action potentials, for example. We talked about the fact that a cell needs to receive neural input in order to fire an action potential. This is not the case. So these specific types of cells, they don't need to receive any neural input. They don't need to receive a neurotransmitter. They don't necessarily need to receive any signal from the outside of the cell. They can, although they can receive signals and result in contraction that way, they also can depolarize spontaneously. So a spontaneous depolarization happens in the absence of any neural inputs, any hormonal input, although these cells can still contract with the presence of that neural input. The innervation is only to a few cells. So again, only that top layer is receiving innervation. The others are kind of just following because of communication through the gap junctions. And because of their spontaneous depolarization, they can exhibit a level of tone. So a level of tone is a contraction that's happening without stimulation. And this is usually seen by our smooth muscle cells that line the vessels. that lines our arteries, arterioles, and to some extent our veins and venules. And so what's happening is the muscles will exhibit this natural sort of general state of contraction. It's not super contracted, but it's also not relaxed, and that is called tone. And that happens in the absence of any type of external stimulation. So if you did nothing to a smooth muscle cell, it would always just have a small bit of contraction, which is called that tone. We can also see increases and decreases in tension, so we can regulate how much force a muscle produces, depending on how much neurotransmitters release, etc. So kind of similar with skeletal muscle, we can increase the force generation for more stronger contractions. We can decrease the force generation for softer contractions as well. There's also the stretch reflex that's exhibited here with single unit smooth muscle. So prolonged stretch results in relaxation, right? Sudden or prolonged stretch induces relaxation of that muscle. That is to protect the muscle. So if there's a lot of blood coming through a blood vessel and there's some stretch happening, we're going to want to allow for some slight relaxation to accommodate more of that flow. Again, a protective mechanism so that we don't injure the vessel, we don't injure the smooth muscle, and we can accommodate different amounts of volume depending on how much stretch is happening on that smooth muscle. There are also graded contractions. So there's no recruitment, and that is because all the cells are already contracting. We'll see later on that multi-unit smooth muscle does have recruitment. But for single unit, because everything's already contracting, right? Everybody's already singing, holding hands, if you will. There's no one else to recruit. Everyone is already, contracting. And so they're acting as a single unit, and so there can be no recruitment. The graded contractions are going to be determined by the amount of calcium. So the amount of neurotransmitter that comes in will dictate the amount of calcium that actually comes into the cell or from the SR, and that varying amount of calcium will also dictate the strength of the contraction, in other words, the strength of the cross-bridge cycling and how much force can be generated. All right, moving on to multi-unit smooth muscle. So multi-unit smooth muscle, typically located in the large airways, whereas single unit is going to be the smaller airways, also in the arteries, so larger arteries, also in the eyes, so ciliary muscles, the iris muscles are usually multi-unit smooth muscle. These have fewer gap junctions, so there are one or two, but they're not a lot, right? And the gap junctions do not have an important function in this type of muscle because each fiber is acting independently. So each one of these smooth muscle cells is behaving autonomously, it's regulating its own contraction, it's receiving its own neural innervation, and it doesn't have tone, it doesn't have that spontaneous ability to depolarize in the absence of any external stimulus. It can, however, exhibit recruitment. So because each cell is contracting independently, if I want to generate more force, I can just recruit an additional cell to provide more cross-bridge cycling, to provide more tension. And so I can have a few cells contracting, and as I need more force, I can call on more cells to begin contracting. So because multi-unit smooth muscle does not act as a single unit, it can recruit additional fibers, and we do see recruitment. All right, here's another just a little illustration that I made here, just kind of ways to classify these two types of muscle. I will capture this here and provide this as well. Okay. Just to reiterate what's going on here, we're looking at the single units up here. Lots of gap junctions, lots of opportunity for communication, the postganglionic neuron only coming into contact with the first layer of cells, and then the signal can spread really quickly throughout. Locations, we said, small blood vessels, gastrointestinal tract, tissues that function synchronously together. So usually in a small artery, all the cells are kind of typically doing the same thing. In larger arteries, the cells may be doing different things depending on signals that are coming in. And so usually in cells that we want to be functioning as a single unit, we have this type of single unit smooth muscle. Other things that we see exhibited here are tone, so the constriction without any input. This is called pacemaker ability. We'll talk more about that with cardiac muscle as well. that also exhibits pacemaker ability. And the third feature here of single unit is that it does not exhibit recruitment because there are no additional cells to recruit. All right. And just showing you what that looks like here in terms of how the actin and myosin are coordinating with each other. And then the spots here where we have the myosin, which is where we see those dense bodies kind of anchoring those filaments and providing for the shortening and the contraction. Contrasting that to multi-unit smooth muscle. So no gap junctions, no tone. We do see recruitment, larger blood vessels, larger parts of the airway and the respiratory tract, as well as parts of the eye and the iris, the ciliary body and the iris. These are areas where we want individualized movement. So think about the fine functions of the eyes. So you may have one eye doing one thing, you may see one part of the eye doing something specific. And so the smooth muscle that controls the ciliary bodies, the iris, they are multi-unit. So they're very individualized cells. Same thing with the larger parts of the airway, larger parts of our blood vessels. Okay, and lastly here, the pacemaker potential that we see with smooth muscle only happens in single unit. We're also going to talk about it because it does happen in cardiac muscle as well. These are basically slow wave potentials. So they're basically cyclical changes in the resting membrane potential. So essentially we start out with a resting membrane potential, which is about -50 for a smooth muscle cell. And then we can change that. So we can change that level with these spontaneous changes in the membrane potential. And if one of these spontaneous spontaneous changes in the resting membrane potential gets us the threshold, we can then spontaneously fire an action potential. Okay, so the slow wave potentials is what gives us the ability to have a pacemaker potential because we're fluctuating this membrane potential. If we had a large enough of a fluctuation and we passed that threshold, then we would definitely hit a pacemaker potential, which is a spontaneous depolarization or a spontaneous action potential. Okay. Just to introduce cardiac muscle here before we end off today, and we're gonna make this a perfect segue into the cardiovascular system. So some of the properties that we'll introduce here of cardiac muscle is that it is similar to skeletal muscle in that it is striated and it has the sarcomeres. So it has those parallel, elongated, striated structures that give for that appearance. It also has troponin and tropomyosin. All right, which helps to regulate the contractions there with the presence of calcium. So very similar to skeletal muscle. It's got gap junctions, which are found within the intercalated discs. We'll talk about those structures next time we meet. It's got pacemaker potential, so cells that can spontaneously depolarize. And then it also has calcium coming from the external environment, so the extracellular fluid, along with the sarcoplasmic reticulum as well. So both sources of calcium, very similar to smooth muscle. Action potentials here last almost as long as the entire tension production. So there is not the ability to sum cardiac muscle contractions because the contractions here last the entire length of the full contraction. The action potentials last the entire length of the full contraction. There is no way for another action potential to come in and stimulate another contraction. So we don't see summation here as well, or what we saw in skeletal muscle. And that is because of those really long contractions, not long contractions, excuse me. That is because the action potential is the same length of time as the tension production, okay? So those are the properties that we see for cardiac muscle. Just to compare here, the refractory period. So this is the twitch or the contraction period of a cardiac muscle cell. This is the length of the action potential in a cardiac muscle cell. Same exact time. So there is no opportunity for another action potential to come in before this contraction is completed, right? So that makes it so that we aren't able to sum contractions here because the action potential is the same length as the contraction. All right. Now, this is a really excellent study tool here. This is comparing features like the skeletal muscle with features of single unit smooth, multi-unit smooth, and then cardiac at the very end. So this is a great way to compare and contrast. Again, that is how you're going to see questions asking you what muscle has this feature, right? What muscle has, for example, gap junctions, right? So we know cardiac muscle has gap junctions, single unit has gap junctions, multi-unit doesn't, skeletal muscle doesn't. So be familiar with this table, be able to recreate this table from your understanding. Another way that I think is useful is to create a Venn diagram. So I usually create a Venn diagram which shows the unique features to each category. So what's unique to skeletal, what's unique to cardiac, and then where some of these different muscle types have shared properties as well, right? So for example, striations are only seen with skeletal muscle and cardiac muscle. So there are ways in which cardiac muscle and skeletal muscle are more alike. There are ways in which cardiac muscle and smooth muscle are more alike. And just showing that in a Venn diagram could be a good way to kind of organize this information.